All right, I think we'll get started. My name is Professor Ken Christensen. I'm the Associate Head for Mechanics Programs in the Department of Mechanical Science and Engineering. And I'd like to welcome you to the second installment of 2013-2014 Disquisitiones Mechanicae seminar series. Disquisitiones Mechanicae means investigations in mechanics, and the notion of this series began in 1997 in the then Theoretical and Applied Mechanics Department here on this campus. At the time, it was held as two-day symposia, one in fluid mechanics, one in solid mechanics, bringing in eminent scholars in those particular areas to campus to give presentations, discuss, and look at cutting-edge aspects of these particular areas. This year's Disquisitiones Mechanicae seminar series is bringing scholars in the areas of biomechanics and mechanobiology. And the idea is to have them visit our campus for 2 days, today and tomorrow, give a perspectives lecture, which we will hear here in a moment, and interact with faculty and staff, and then give a more focused scientific talk on Friday, which I will let you know is happening tomorrow at noontime if you're interested. So with that as background, I'd like to introduce Professor Tahir Saeed from the MEXI department, who will introduce today's speaker.
Thanks, Ken. It's my pleasure and honor to have today with us Professor Paul Jenny from University of Pennsylvania. So Paul asked me to keep it very short and give more time for technical discussions. I will do that. Professor Jenny graduated in philosophy from Oberlin College in 1976. And then he did his PhD in University of Wisconsin-Madison in chemistry, focusing on polymers, physics of biopolymers. And then he joined Harvard Medical School in 1982 and worked as a research fellow and then as an assistant professor and associate professor in Harvard Medical School until 1999. And he joined the Penn in 1999, and since then he has been there. And as a professor of physiology, and also in the departments of physics and astronomy, biochemistry, and cell and molecular biology, cell and developmental biology. And he did extensive work on viscoelasticity of biopolymers, rheology of biopolymers, intracellular, extracellular, worked extensively on the mechanics of cells, pathology of cells in relation to the mechanical properties, and the relationship with extracellular matrix. He got quite a few awards, including Common Fellowship as well as Curie Fellowship. So he's very well known internationally as well. But what doesn't show up in his CV, I think I have to mention, that I think that his works, his write-ups over the last couple of decades have actually inspired a whole generation in cell mechanics. I'm one of those beneficiaries. So many of the people that he has talked with have already told me that he has been inspired through his earlier work as well as the discussions that he has had today and motivated more as well. So in a way, he laid some of the foundations of cell mechanics, and that's a significant part of his contribution. And some of his works are now part of textbooks on cell and development biology. So the contributions are enormous. So with that, I would invite
Hi, thanks very much. Can you hear me okay? So great. Thank you, Terry, for the kind introduction and also the chance to come back to this really, really wonderful campus. It's great. It's my 4th or 5th time here. I really love it. Okay, so I want to tell you a couple of stories about what we've been working on recently to try to see whether mechanical signals. Did I steal the laser pointer? It's not in my pocket. Oh, there it is. Sorry. of how the physical properties of the environment impact cell biology, and to see to what extent does that sort of mimic or reproduce or alter the way the chemical signaling works. So I want to show you a couple of stories about how changing the mechanical properties of the materials in which or on which cells bind changes their own mechanical state, but also things like their morphology, their cytoskeletal assembly, and functions, things like proliferation. So the work I'm going to show you is done by engineering postdocs and grad students in the lab, Anand Chopra, Justin Weiner, and Jeff Byfield, and a cell biologist, Maria Murray. And we also, towards the end, I'll show you some experiments done by a really fantastic physics student who's visiting us on a Fulbright, Tasha Pagoda, this summer. Okay, so here's the kind of outline of the three stories I'd like to tell you. One is to sort of show the evidence that has accrued over the last few years that suggests that the physical properties of things like soft tissues and organs are just as important, or at least vitally important for the eventual biologic or physiologic outcome of the cells, and that they can provide very specific signals that can be just as strong and specific as chemical stimuli. And part of the specificity by which physical properties can impact the cell depends on the fact that different cell types are surprisingly differently sensitive to physical cues, by which I mean imposition of forces from the outside, or by encapsulating cells on stiff or soft materials, or purely elastic or more viscous materials. And one of the reasons that cells are so specific or so different from each other is that the mediation of this physical information comes through specific transmembrane receptors in exactly a parallel way to the way that hormones and acute biochemical signals impact the cell by binding to some receptor. What those receptors are for physical cues are much less well worked out, but that's not really surprising. People knew that insulin impacted glucose metabolism for decades before anybody knew what the insulin receptor was, and we're sort of at the similar state here for physical cues. But what I do want to show you a couple of examples of three different classes of transmembrane receptors that mediate this kind of physical interrogation. The best studied a class of receptors, much of which has been done here, has been done looking at how integrins, cell matrix receptors, do this kind of mechanical signaling, but also cell-cell adhesions through cadherins are also equally important. And I want to show you towards the end what happens if you take a whole different class of receptors for an extracellular matrix component called hyaluronic acid, and show you that this very much changes how mechanosensing works. And underlying these kinds of questions is always this notion: can physical stimuli be mimicked? Are they different in kind from the way that biological stimuli work, or do they activate the same pathways? And if they activate the same pathways, can we play around with the responses to physical cues by getting the biochemistry inside the cell appropriate? Okay, so just as a way of introduction, here are the kind of the tools of the trade for what in our case are fairly simple measurements of either tissue mechanics or single cell mechanics. So a very commonly used way to measure in the bioengineering literature and elsewhere is to do to a piece of tissue or a macroscopic piece of material the same thing that polymer physicists do to, or material scientists do to chewing gum, bread dough, everything else, by putting it in between two plates, applying a torque to one of the plates, either oscillating or static, and looking at the deformation, and from the relation between the applied stress and the resulting strain, calculate a shear modulus. And I'll talk a little bit more about this tomorrow, but it turns out that there's a very interesting interplay between doing these kinds of shear rheologic measurements and applying uniaxial steady stresses in the orthogonal direction. So this is something that doesn't automatically happen with simple elastomers or polymeric materials, but for biological materials, this turns out to be a big thing. The other kinds of tools of the trade, rather than putting a torque, a shear strain on a macroscopic piece of material. It's also very easy to indent materials with a flat indenter. This is measuring the stiffness of the material exactly like what you would do if you poked somebody with your finger. You could more or less figure out the softness of the thing that you're touching from the same thing that one of these vertical probes does. Figure out how much force and how much force you apply and how much indentation that results in. And the kind of the nanoscale variant of these uniaxial indenter devices is the atomic force microscope, and the same principle applies, and this is roughly drawn to scale. The cell measurements I'll show you later on have been done by gluing about a 2-micron sphere to the AFM cantilever and gently poking a few hundred nanometers into the top of a cell to see what its viscoelastic response is. Okay, so if you do something like that and take a very, very crude approximation and for a moment completely forget about time dependence or strain dependence and simply take a lot of tissues and look at their elastic modulus, in this case converted to a shear modulus, and a small strain limit on a time scale of about a second. A lot of interesting comparisons come out between tissues. I'll show you a very small subset of this, but it turns out that in normal physiology, the elastic properties of a normal healthy tissue from the same animal, from the same age approximately, it's a remarkably well-controlled parameter. So I don't have error bars here, but here, for example, are the variances of measurements of many, many different aortas that are either normal or disease. And you can see that the variance in the normal state is surprisingly small compared to the mean. So the stiffness of organs is not an accident of what the cells inside are doing. It's a very, very well-controlled parameter for whatever reason. And if you look at the disease development in many different contexts, so for example, the development of fibrosis in the liver, the elastic modulus of a normal human liver or a normal animal liver is a few hundred pascals. That rises approximately in order of magnitude, even fairly early during the development of fibrosis. The most thoroughly studied example of this is probably development of malignancy in the breast, where the stiffness of breast tissue increases quite a lot, even early in a fairly pre-malignant or histologically pre-malignant lesion, but becomes much stiffer as the full-blown tumor arises. And this, of course, is why palpation works as a diagnostic method. But these kinds of stiffness differences arise in all sorts of pathophysiologic states. And one example that I highlight here is work from Rick Asoyan's lab at Penn, in which we've looked at the elastic moduli of aortas taken either from normal wild-type mice or mice that are genetically depleted of ApoE, a component of a lipoprotein complex. But in any case, loss of this particular protein predisposes these mice to getting atherosclerosis. But even before they have any sign of the disease, if you sacrifice them before there's any kind of histologically evident lesion in their blood vessels, they're already stiffer to begin with. And so this, as well as this stiffening of the fibrotic liver, suggests that stiffening of a tissue might be at least a contributing factor, and I'll show you some evidence for this in the next slide. It's not simply a consequence of the disease, it might actually contribute to its development. But I don't want to overstate the case. Stages in stiffness aren't simply a pathologic thing. During normal embryonic development, there's an enormous change in many soft tissues, right? Embryos are much softer than adults that they generate. And one of the more spectacular examples is the stiffness of the embryonic heart, or the cardiac cushion from which the heart cells will eventually arise, is orders of magnitude softer than the adult heart that it will eventually generate, and just as a normal part of development, and I'll get back to this in a moment. It's also not true, I'll talk a little bit more about this tomorrow, stiffness of tumors are not always stiffer than normal tissue from which they arose. And one of our projects recently has been to look at the mechanical changes in brain tumors. And somewhat surprisingly, the elastic modulus of an excised glioma tissue is no different from that of a normal piece of brain. So it's not an automatic occurrence that the tumor is stiffer than the tissue from which it came. But why that is something maybe we'll touch on tomorrow. Okay, so here's a little bit of the tools of the trade of how we have been trying to measure in many labs, and I've been been trying to measure the response to these mechanical cues in a kind of a controlled way. And the big tools of the trade are these two things. Well, almost all the data I'll show you in the next few slides come from our attempts to use basically a method developed by Yuli Wang about a decade and a half ago now, in which you simply substitute the glass cover slip on which traditionally cell biologists have cultured cells, and you put on top of them a soft cushion. It's about 100 microns thick, is optically transparent as much as you can make it, and in one case, in the simplest case, is chemically inert to the cell, meaning the cell, if you simply put it on top of this cushion, would not be able to bind to it because it has nothing sticky to engage. In order to get the cell to stick, you covalently put on top of this thing whatever you want, fibronectin or collagen or some kind of ligand that mimics the extracellular matrix, or you could put a fragment of a cell adhesion molecule like a cadherin to make the cell bind to this soft cushion as though it's binding to a giant flat cell. But you can put pretty much whatever you want on there. The variation I'm going to show you is what happens if we substitute the inert gel, which in most cases is polyacrylamide, by another semi-inert gel made out of cross-linked hyaluronic acid, another very flexible, linear elastic hydrogel. It's very well studied. It has the slight difference that it's a polyelectrolyte, but doesn't matter so much for that. But most cells will not bind to this by itself, nor will they bind to polyacrylamide. So in order to get the cells to stick, we still have to put fibronectin, collagen, or something. But the difference is that this turns out to have, cells have receptors for this thing, even if they can't anchor to it. But you can make now you can make all sorts of objects work and change the elastic modulus from maybe 50 Pascal to 50,000 Pascal, and so span the whole range of tissue stiffnesses that I showed you in the earlier slides. So we can mimic things like embryonic tissue on the soft side to things like cardiac muscle and stiffer things. We can't quite get to the stiffness of cartilage, but we're kind of on the way there. The other system, which I won't have time to talk to you about, but it's actually equally revealing and also very commonly used, is rather than make a continuous hydrogel, make little soft pillars made out of PDMS, which has an elastic modulus that's very high, off scale of the stiffness of what you can make with hydrogels, but you can make these little beds of nails effectively soft by making them slender and tall. And just from the deflection of the tops of the pillars, the cells can either move them easily or move them hard. And it turns out that this is a spectacularly good mimic of making a soft material. I just want to take a minute to point out how surprising this is. Why should a cell care if a PDMS pillar is tall rather than short? Because the material to which the proteins adhere is still almost infinitely stiff for the cell. So every integrin or every adhesion molecule that lines the top of one of these pillars, and these pillars are typically a couple of microns in diameter, so hundreds of adhesion molecules bind there. They don't care about the local stiffness of the material to which they're actually attached. What the cell really cares about is the work it has to do to deflect one pillar top with respect to another. So we're using these things to try to figure out the length scale, the size of an object that a cell can measure the stiffness of. Evidently, it cannot detect, or it doesn't care that this is stiff. It just cares that the top of the pillar is flexible. But that's just an aside. I want to mostly show you things about these hydrogels. Okay, so here's a relatively typical experiment using hepatic stellate cells, the cell type that causes fibrosis in the liver to become accelerated. So this is a cell type that will spit out extracellular matrix, inflammatory signals, and really drive the fibrotic disease. So if you isolate a hepatic stellate cell from a tissue that's either normal or stiff, the environment that it comes from turns out to be quite different. So for example, if we measure the stiffness of a liver, of the whole liver, the environment in which these stellate cells will live as a function of stiffness, the stiffness of the organ increases relatively quickly after we've caused the fibrosis to occur. In our case, we cause the fibrosis to occur by intoxicating the animals with carbon tetrachloride. So the animals will be doomed to get fibrotic And if we sacrifice them and look at the stiffness of the liver, it's increasing a few days after the initial intoxication. But if we look by histology, if we send slices of this tissue to a pathology lab and ask, does any cell in there look fibrotic-like? The answer is no, until a couple of weeks after the injury has occurred. So the stiffness actually precedes the cellular dysfunction or the cellular morphology changes that take place later. What we think is happening is the stiffness arises not because cells are changing their structures yet or laying down more matrix. They're changing because lysyl oxidases are activated. The matrix that's already there is just more heavily cross-linked, and the cells will react later. The way they react is shown here in a kind of in vitro mimic of the liver stiffening I showed you before. This is work done with Rebecca Wells's lab at Penn. So if you take a hepatic stellate cell from a healthy liver that has a very small elastic modulus and put it on one of these hydrogels coated with fibronectin, a hydrogel that has the same stiffness as a healthy liver, these little cells will bind and they're metabolically happy, but they're not photogenic looking cultured cells. They're slightly brown because one of their functions is to store lipid soluble vitamins. They're kind of translucent, but they don't spread very much, but that doesn't mean they're not healthy. They're actually perfectly healthy. As we change the stiffness of the material underneath them, but don't give them any chemical stimulus that we know of, simply changing the elastic modulus underneath is enough to cause this massive, it causes proliferation, which I don't show you, but the cells become much, much more stellate. They elaborate this very big cytoskeleton. They start to express extracellular matrix, and they drive all the fibrotic pathways that will eventually lead to disease. But we've triggered this not chemically, but simply by playing with the same range of stiffness that occurs ordinarily during fibrotic development. I show you a couple of slides on a different cell type, on cardiac myocytes now, to make the point that these mechanical stiffness responses are not the same for every cell type. In fact, they're not even always monotonic. And the most dramatic example of cells having a very small window of stiffness in which something interesting occurs happens in muscle cells. Several groups have looked at skeletal muscle in the past. We have a collaboration with Yasha Kresh's lab to look at neonatal rat ventricular myocytes. So taken out of the healthy beating heart and placed immediately onto gels that are either much softer than the tissue from which they came or much stiffer than the tissue from which they came. The cells will bind to these soft or stiff matrices perfectly fine, and they're metabolically okay, and they actually look pretty good, but they do not look like the cells in your heart should look. If instead you plate them on chemically identical gels that have the same elastic modulus from the tissue from which these came, they immediately form, or not immediately, but spontaneously regenerate this elongated sarcomere-containing shape that they had in the tissue from which they came, and they begin to beat and function normally. And you can accomplish this not by patterning these asymmetric shapes, but simply by putting them on mechanically realistic things and letting the cell do its own thing. In this case, we're putting them on gels that are coated with a ligand for integrins, so all of the mechanical signaling in the adhesion is done through integrins and all this army of accessory proteins that link the transmembrane protein complex to the cytoskeleton. But if we do something very different, we substitute a cell adhesion molecule, N-cadherin, for the integrin ligand, something very similar happens. So in this case, we're not putting the cell is not able to engage any of its normal extracellular matrix or integrin receptors because it has nothing on the soft hydrogel to deal with. It's only engaging through cell-cell contacts, and yet they can do a very similar thing. If the elastic modulus is too low, nothing, you get these small amorphous cells. If the elastic modulus is approximately right, they don't do the job quite perfectly, but they come pretty close. So everything from the signaling point of view, things are very different if you engage things through cadherins rather than through integrins. But still, this kind of elongated shape arises only if you get the elastic modulus appropriate. If you get it on the high side, the cells will still live, but they'll become very fibroblastic. So this seems to be a very common signaling pattern for many cell types through a whole large class of receptors. I'll show you one other example to emphasize that this is not simply a kind of a morphologic phenotype of the cells, but many functions and many transcriptional programs are also very strongly dependent on these stiffness cues. And one of the more interesting effects of playing around with matrix stiffness is changing the cell cycle. So in many primary cells, you can block the cell cycle at various points in its, either in G1 or in G2. You can stop the cell cycle and the production of cyclin D, which the cell needs to undergo cytokinesis and mitosis. You can block it simply by putting it on a very soft substrate, and not a pathologically soft substrate, but a substrate that is approximately the same as a healthy organ in which cell proliferation in an adult is relatively slow. So if you do that with human bone marrow-derived stromal cells, and you put them on a matrix that's very soft as the bone marrow in which these cells normally just sit, but don't necessarily proliferate, they will sit on these materials for a very long time, and they will not divide. So BRDU incorporation, a measure of entrance into S phase, is very low on these soft materials. If you make the elastic modulus a little bit bigger, they begin to proliferate. They proliferate almost as much as they do if you put them on glass. Pretty much everything is going to proliferate if you make the matrix stiff enough, but we haven't done anything chemically. And I'll show you here as an example of how big this cell cycle block is. You can reduce the amount of cell proliferation by changing the elastic modulus at least as efficiently as you can by making them confluent. So this stiffness the stiffness influence on the cell cycle is just as important as contact inhibition for many cell types. But these cells, just to show that they are still viable cells, we haven't made them unhealthy or incapable of functioning later by growing them in a soft environment. Jessamine Weiner did this experiment using a method of Beningo and Wang to make a little sandwich. And what she's done here is done what I showed you here, incubate some cells on a very soft substrate so they're quiescent for a while. And then on day one, take two of these kinds of little round cell populations and put on top of them a little hat. And the hat is made out of either a hydrogel that is just as soft as what they were sitting on, or something that's very stiff. And if you put on the top surface something that's very soft, the cells will not proliferate for a couple of days, as long as we can keep them healthy. So this field of cells is the same cells that were here 2 days ago. They've changed their shape because they're adhesive on top and bottom, but they have not proliferated. They haven't even moved. If instead you put them on top of them something rigid, you'll see that these things have very rapidly changed their shape, begun to proliferate, and eventually, in all cases, we can get them to become whatever cell type we want by playing around with the induction medium. In this case, we've made them become osteocytes, a cell type that wants to see a hard substrate. Even though they've been sitting on these soft materials for a long time, they do not lose the genetic information they need to become a differentiated cell. Not only do they change their function, but the cell is also changing its own stiffness. So a peculiar aspect of many cells is that they will remodel their cytoskeletons or their cortical contraction in order to approximately match the stiffness of the material under which they are put. And the bone marrow stem cells are particularly good at that. They will undergo a kind of what looks like a biphasic change in their own internal stiffness, even without any kind of differentiation down a particular cell fate. So just an induction medium, these cells will become stiff if we put them on stiff things. They will stay soft if we put them on soft things. And as they sit on soft things, as the Disher lab and many others have now shown, if you culture them on soft materials, these cell types like to differentiate into different differentiated cells which are soft, like fat. They don't want to become cartilage, which is a stiff cell, but if you kind of pre-incubate them on stiff materials, they're ready to go and respond to differentiation media. Whoops, I hit something wrong. There's also a very interesting difference between normal and cancer cell types. I'll talk a little bit more about this tomorrow, but if you take a glial cell like an astrocyte that again changes its stiffness in response to substrate stiffness over a range that's approximately relevant to central nervous system tissues. If you take glioma cells that arose from the same precursors of the astrocyte but are now transformed, they do this very, very differently. So they're much softer, they're much less mechanoresponsive. They still are mechanoresponsive, but they've lost a little bit of this control. So let me conclude this first part by pointing out that soft tissues have a surprisingly well-controlled elastic modulus. And that modulus changes with disease state and development. But the cells within that tissue do not have a pre-programmed stiffness. An individual cell can change its own stiffness over a very large range in response to the change in the mechanical properties of the stuff around it. The way cells do this is very cell type specific. Different cell types respond very differently, and that response depends on the kinds of adhesion molecules it uses to engage the matrix or the material in which it lives. And what we've been interested in lately, and I'll show you some examples, is we're trying to understand how this, two things. One, how does the stiffness change during development and disease? Is it the cell that's changing and changing the matrix, or is something else changing the matrix and the cells are responding differently? And the whole issue of whether in a big composite, complex material like a tissue, how much is cell and how much is matrix is still very much an unresolved thing. But the other question that we're trying to understand is how does the cell, what are the molecular mechanisms that allow the cell to sense things like matrix stiffness? That in the very simplest case, you can imagine that there is a whole series of small molecular springs that come all the way from the nuclear matrix to the nuclear membrane, through link complexes or something, through the cytoskeleton, to proteins on the intracellular side of the transmembrane proteins, to the extracellular matrix proteins, which are also large, flexible molecular springs. And if you now put attention across this whole bunch of springs, you can sort of imagine the two scenarios. If the cell is relatively rigid and the outside world is very soft, if you put attention across this whole stream of molecules, the one that's going to unfold the most is the softest. So if the outside world is softest, you might unfold something, expose a cryptic signal that will signal to something else in the cell and then drive things just like they always were driven through chemical signal transduction pathways. Or it might pull on an ion channel and again elicit basically biochemical signaling, but through an initially physical event. It may also be much more physically intact. It could be that you transmit the stress all the way from the outside world to something either inside the cytoskeleton or maybe in the nuclear matrix itself, and then drive things very differently. So the question is whether mechanical signaling is fundamentally different from chemical signaling, or can it be bypassed if we knew what the biochemistry was in more detail. So here's a couple of examples of, I'll go through these relatively quickly because I don't want to dwell on the molecular mechanisms too much. But there are two classes of fairly commonly observed changes that cells undergo when they experience changes in the rigidity of the thing around them. So there are things that happen fast or acutely, and there are things that happen very slowly and that are adaptive and transcriptional. The things that happen really fast are really of three kinds. One is that someplace on the nuclear membrane are changes in the activity of things like P120 Rho gap, which is a gap for, not surprisingly, Rho, but other small GTPases. So if you inhibit this activity, you upregulate Rho GTP activity. You also upregulate some transcription factors that seem to be specific for endothelial cells. But you go through a lot of molecular signaling pathways that will activate things like Rho-dependent kinase, that will activate myocyte light chain, that will increase contractility. At the same time, rigidity also increases things like the phosphorylation of focal adhesion kinase that will drive a different class of small GTPases that will drive actin assembly, lamellipodial formation, all kinds of cytoskeletal changes, but that will also impact cyclin D and control proliferation. So I already mentioned that stiffness changes a lot the proliferative state, and this is one of the pathways through which it might work, and largely driven by work from here, but from other labs too. It's also an equally plausible thing to think that changes in rigidity that give rise to these changes in contractility of the cell can deform the cytoskeleton in different ways or transmit force directly from the cytoskeleton through mediating protein complexes that link to the nuclear membrane, that eventually link to the chromatin, that might change the disposition of the DNA inside and make transcription events different in response to physical cues. If this is the way that things work, then it's really going to be difficult to reproduce this in a biochemical way, because there's really no biochemistry going on here. It's all mechanics until you get into the nucleus and start pulling on the DNA. But it's clear that a lot of these things do, in some sense, change the nuclear, at least transcription activity. In one way, there are two different pathways through which this might work. One is a more biochemical pathway in which, as I also already mentioned, things like mechanical cues will change the extent of actin polymerization. And there are transcription regulators like MRTF or MAL that are inhibited by cytoplasmic actin. As you cause more actin to polymerize, as the cytoskeleton reorganizes itself in response to rigidity, you lose these actin monomers, you lose the inhibition of the transcription factor, that goes into the nucleus and does what it does. The other way is kind of more fundamentally mechanical. There's also a second class of nuclear transcription regulators called YAP and TAZ, which have gotten a lot of interest lately, in which tension at the cytoskeleton is somehow sensed directly by this transcriptional regulatory complex that now goes into the nucleus and causes a lot of adaptive changes that they're characteristic of cells grown on rigid, maybe fibrotic, maybe cancerous tissues, and not softer materials. So I want to get back to this point later, but I just wanted to give you a flavor of the kinds of things that people have elicited from mechanical changes. But here's kind of what we're more interested in. Most of what people have done in vitro, us included, and the experiments I showed you in the beginning, is to take a single cell, or maybe a small island of cells, and put them on soft hydrogels or pillars or something that have one specific protein that will bind one specific transmembrane receptor so that we can figure out what the molecular ingredients might be. But in real life, this scenario is much more complicated. There's simultaneously mechanical signaling from one cell to another. There are multiple kinds of transmembrane proteins that engage this extracellular matrix that signal very differently. But in addition to things like integrins, which actually can act as anchors, there are a whole slew of proteins, including receptors for this extracellular matrix polysaccharide hyaluronic acid that I'll talk about, that also engage the extracellular matrix. They can't act as physical anchors, but they change the response. And I want to show you a couple of examples of this, which have turned out to be surprising. They're motivated by this problem. I already showed you that if you take cultured cells and put them on very soft materials, you cannot make a functional myocyte. Regardless of what we do chemically, we can't make a cell that looks like this sit on a material that is as soft as this, if we only engage them through integrins. The problem is that this is the stiffness of an adult heart, where these sarcomeres are commonly requiring a fair amount of tension and a large amount of stiffness. But in the embryonic heart, where the sarcomere first formed, that's as soft as this. So how did those early myocytes form if you need tension to get, or stiffness to get them to form? Because they're forming in a very soft world. And that's where hyaluronic acid comes in. This is the most common, this is the most prominent component of extracellular matrices early in development, when there isn't very much collagen fibers, there isn't very much fibronectin yet. This is the world in which embryonic cells originally grow. It's also a matrix component that's very strongly upregulated in cancers. It's upregulated in wound healing. So cells will synthesize and secrete this large polysaccharide and at the same time upregulate receptors for it. And it turns out that hyaluronic acid is capable of doing this. So if we substitute the inert polyacrylamide gel on which we have stuck fibronectin as the anchor to a myocyte, in which case cells look like this, all we've done is substituted the inert polyacrylamide with a almost inert hyaluronic acid gel as the thing that makes the soft matrix, and again decorated with fibronectin, and now we get these massive myocytes. So full of sarcomeres, they're beating, they're really beautiful cells. They do much less work during the contractile cycle because they don't pull as hard, but they have all the contractile machinery they need, and they're adapted to a much, much softer world. And this is not unique to myocytes. So cardiac fibroblasts do the same thing. I showed you before, this is what I just showed you, small myocytes on a soft polyacrylamide gel, beautiful elongated myocytes on HA plus fibronectin. This is what they look like on glass. Fibroblasts do the same thing. I think what we're most familiar with are these beautiful stress fibers of fibroblasts on stiff materials that go away when you put them on soft materials. But if you put them on very soft materials that have a combination of integrin ligands plus something that will bind hyaluronic acid, you regenerate these massive stress fibers, even though it's sitting on something very, very soft. It's also true for mesenchymal stem cells, exactly the same phenomenon. It works with endothelial cells, and in particular, it's not just the actin cytoskeleton and the big stress fibers that reappear. These large focal adhesions, in this case it's a vinculin stain, that are typically characteristic of the edges, the focal edges of a cell that is undergoing a lot of tension, like on a very stiff material. These things also reappear, even though the cell is sitting on something much softer than it needs to generate tension. So we've done a couple of control experiments to make sure we're not fooling ourselves that these gels are as soft as we think they are, and because it's possible that the cell that's sitting on top of the gel is reorganizing the matrix and laying down its own stiff little material. So here's an AFM experiment in which we've poked on the hyaluronic acid gel at various places on this little grid-lined hydrogel, and there's one cell sitting on there. I hope you can see the outline of the cell. The red points are places where we poked with the AFM to measure the elastic modulus of the cell. So here's how stiff the gel around it is. It turns out, in this case, it's about 600 Pascal. The cell sitting on top of it is a little bit softer, but close in the same region. And now we gently disrupt by calcium chelation, pop the cell off of the matrix, and look underneath the cell. What was the cell actually touching when we did the measurement? And it's just as soft as it ever was. So the cell is clearly not fooling us by placing a kind of rigid platform underneath it to which it's sitting. It's actually sitting on something soft. So what the hyaluronic acid is doing is changing the response of the cell to change the set point of stiffness where things begin to form. So things like large adhesive area or appearance of large stress fibers that typically happen on very, very stiff materials, if the only signaling comes from integrins, are happening at much, much smaller stiffnesses if you put this extra layer of signaling on top of it. I'm going to skip over, I'll show you this slide, I'll skip over the next one. One of the things that's been intriguing is that the signals that come from these two receptors, that come from integrins and that come from the hyaluronic acid receptors, they are not independent signals that can be coming from spatially distinct part of the cells. Because if we do the same sandwich assay I showed you before, but coat cells on a very soft fibronectin-coated gel where they're round and amorphous, if all they needed was hyaluronic acid somewhere to now cause them to spread, we ought to be able to accomplish that by putting the fibronectin on one side and putting hyaluronic acid on the top surface of the cell. If we do that, the cell does not spread. If instead we put hyaluronic acid plus fibronectin on top, just as we did before, now it spreads like crazy. So it ignores the soft, round cell information it gets from the bottom. It's now dominated by the let's spread and make a stress fiber information that it gets from the top of the cell. And the slide I'll skip over later simply makes in more detail the point that this is very much integrin type dependent. This trick of spreading for mesenchymal cells works only through fibronectin receptors. If we try the same trick with collagen, so we coat this gel with hyaluronic acid plus collagen, it doesn't work. The cells have receptors for collagen. They'll engage the collagen, but they won't engage the spreading response. So this seems to be not entirely mechanically driven. This is a way of chemically inducing the same response that stiffness and mechanics ordinarily develops. And it's an acute response. The cells immediately know when they have touched a substrate that's either soft or hard or has HA or only integrins. So here, for example, are embryo fibroblasts spreading on either stiff or soft materials as a function of time. And T0 here is the moment that we see a cell from three-dimensional suspension touch the surface of the gel. And if you do it on a stiff matrix, like or on glass, for example, there's typically a couple of 1 or 2 minute period where the cell attaches, figures out what's going, and then immediately starts to spread in a way that looks like the area is increasing approximately linearly for a while with time. And if you put it on a very soft material, the first couple of minutes are very similar, but then instead of spreading nice and regularly, it kind of oscillates back and forth, extends protrusions, pulls them back, and never gets bigger. So if you put them on very, very soft things that have this hyaluronic acid trick, boom, they start to spread immediately. So this is not a transcriptional regulated thing. It's an immediate response that's signaled through one of these acute pathways I showed you at the beginning. I'm going to skip over this and show you that this combination of mechanical cues and the appropriate chemical signaling also overrides this mechanical block of the cell cycle I showed you. So if you look at something like cells proliferating after touching a surface, what I showed you before was that if you touch a cell to a very soft material and don't do anything in particular to it, it will just sit there. The cells will sit, they'll stay alive, they won't proliferate. If you put them on something stiffer like tissue culture plastic, they'll proliferate very nicely. But if you put them on these hyaluronic acid gels, they will proliferate again immediately, even though they're sitting on something soft. So I think what this is telling us is that we're able to somehow bypass the stiffness of materials by doing the right biochemistry. So at least in this case, the mechanical cue is not different in kind from biochemical signaling. It's transduced relatively early into a biochemical signal we can reproduce by something else. I end with one example of our attempt to prove that by traction force mapping that these cells are able to make these massive actin bundles that look like stress fibers without actually applying tension to the cell. And the way we've done this is a nice trick that Anand Chopra learned how to make. It turns out that the chemistry of making these hyaluronic acid gels is very amenable to micropatterning. So what we've done is micropatterned, not the hydrogel, but micropatterned a glass surface with little dots of fluorescent fibronectin. So the only molecule that the cell will be able to make contact with. So we pattern a glass surface with 1 micron beads of fluorescent fibronectin spaced about 10 microns apart. And then this is upside down from the way we actually make it. And then we pour the gel on top of it. And during the chemistry of gelation, the cross-linking moieties will capture the fluorescent fibronectin, bring it to the top of the gel. And now we can have 1 micron dots of adhesive proteins on an otherwise inert hydrogel spaced however far apart we want. This works much better than trying to modify the gel itself. Then we put a cell on top, and here's what it looks like. So we can print square grids of these 1 micron dots out of gel that's less than a kilopascal stiff, and put a fibroblast on here, which ordinarily would not make stress fibers until the elastic modulus was 50 times bigger. But here it's making them immediately. So we get big actin bundles and stuck on, the only place the cell can adhere is on these little red dots. And we've been trying to ask, well, okay, how much force does the cell apply to these dots? And this is a pretty easy thing to do, because here's another kind of more contrasty picture of the square array of dots that we printed on there. And you'll see that the cell makes many nice adhesions to these dots that have this kind of curvature on them that looks as though there's tension applied to them. But because we know the elastic modulus of the gel and the height of the gel and how big these things are, we can simply measure the displacement of the gel that the cell, the displacements of the dots that the cells are, quote, pulling on away from the square pattern in which they are put, and simply take, oops, I keep hitting this inappropriately. We trace out the little circles where we know the cell has touched it, and instead of removing the cell through trypsinization, we just kind of don't look at it optically anymore. We just get rid of the green channel and you just look at the red channel where the dots are. And you'll see that some of the dots are actually kind of deformed a little bit by the integrins that have touched it. But if you look at the displacement of the circled dots, are the ones that were at the edge of the cell, that were at the end of one of these actin bundles, they have moved remarkably little away from the pattern on which they were stamped. And so you can calculate from the displacement on the top surface of the gel and the elastic modulus of the gel, you can calculate the amount of force that's put on it. The displacements are always submicron. On a gel that's maybe 100 microns thick. So we can calculate approximately the force that's put on there, and it's a remarkably small force. So if you calculate the stress on one of these little dots, it's less than 50 Pascal over a dot that's about a square micron or so. So that's less than 50 piconewtons of force over an area of contact that has hundreds of adhesion molecules on it. So remarkably little force transduced into the network, and yet we're making all the ingredients that are typical of a stressed cell. So I'll conclude that. I'll skip over the signaling, which we really don't understand. We're just beginning to try to understand it. I'll just tell you what the punchline is. But what it looks like is the substrate mechanics is a very strong control of cell structure and function, proliferation, many functions. And it's been it's exaggeratedly strong when signaling goes only through a single class of receptors, through integrins or cadherins. If that's the dominant attachment site, the mechanics seems to be overwhelmingly important. However, if you start playing with other chemical signals, in particular signals from hyaluronic acid receptors, in concert with signaling from integrins, you can do a lot to change the set point of the mechanical cues that a cell needs to perform some function. And in particular, cells on these very soft gels are able to make very large adhesion sites and actin bundles, the kinds of things you typically associate with tension, but the cell itself is not applying tension, at least not to the substrate underneath. How this works, we totally don't understand. We're beginning to fish out some of the ingredients that this is using. It seems to be particularly receptive to signaling through alpha V beta 3 integrins. Other integrin types don't have such a strong effect. It works through at least two HA receptors, CD44 and CD168, and a lot of the common pathways I showed you before, Rho kinase pathway, but also phosphoinositide synthesis, and probably many other biochemical signals are yet to be determined. Okay, thank you very much for your attention.
Oh, that was an awesome talk. A little overwhelmed, I'm not sure where to start. Do you have any speculation that you can give with respect to this? I know there's a lot of signaling pathways that you're looking at, but the fact that they're making these focal adhesions on the soft gels, what role is hyaluronic acid playing? Is it guiding the formation of these structures? I think it's just really surprising.
Yeah, it's a little surprising to us too. I think what there are two things we're trying to chase. One is hyaluronic acid receptors engage a class of cytoskeletal proteins called ERM proteins, so azirin-dixin, things that are not typically part of the big focal adhesion, but they still have a big role to play in stabilizing the actin cytoskeleton. So that pathway could be acting synergistically with whatever the integrin is doing. We still need integrins to get these big focal adhesions, but whatever the HA is doing is sitting on top of that. The other thing that HA does that is much less well understood is it stimulates the synthesis of phosphoinositides. So especially the synthesis of PIP2 from PIP and PIP3 from PIP2. And those phosphoinositides are also big drivers of actin assembly. And they will activate things like tailin, they'll interact with vinculin. So many focal adhesion complexes are also drawn to those lipids. So we're wondering whether that lipid synthesis isn't part of what's making these adhesions form.
So when you put it on polyacrylamide gel, have you done this before?
Yeah.
You don't see the formation of the
No.
focal adhesion?
No, it's much harder to make such a good square array, but they will stay round on the polyacrylamide.
Very nice talk. So one thing I wondered about is, when you look at the differences in the tissue responses to mechanics, how much would you speculate on how much that's actually due to differences in receptor expression?
Yeah.
So, do you see this hyaluronic acid response, for example, in a difference in other tissues?
Yeah. Yeah, so far I think we have not seen cell types, at least primary cells, that do not have some kind of enthusiastic response to hyaluronic acid, but it only works if you couple it with the appropriate integrin. So it does depend, and one of the things that Kasia Pagoda worked on that I might talk a little bit about tomorrow is that we've looked at 3 different human glioma cells. They all will respond to hyaluronic acid because hyaluronic acid is also very prominent in brain tumors and many other tumors. But these cells express different subsets of integrins. And depending on which integrin type, some will get turned on by HA plus laminin and they won't care about collagen, and the other ones are exactly the opposite. So the receptors that different tissue cells express that spectrum is going to be really important for how they mechanosense, I think.
When you have these very soft shells, very often they not only deform in the plane, but also deform in the all-plane direction. How much of the three-dimensional effect?
Yeah. So as far as I know, in these little stamped HA gels, we see, two things. In the stamped HA gels, we see very little out-of-plane motion because we see very little in-plane motion too. But if we take a cell that pulls stronger, like a mesenchymal stem cell on a fibronectin-coated polyacrylamide gel, there the out-of-plane motions can be quite big. So it looks to us as though the edge of the cell, if it's contracting hard, the edge of the cell is pulling up and the center of the cell is pushing down. It's either pushing down or it's just passively has to go somewhere. And those movements are on the same order as the transverse movements. They're smaller, but they're still surprisingly big. And if we put cells on something like a collagen gel or a fibrin gel, where we can put little fluorescent nanobeads on the fiber, then things go really bad because I think because the architecture of the networks is so strange and they have this negative normal stress when you put them in shear. As the cell pulls this way, the beads actually go out of focus even in a not very focused microscope. So there's a great deal of downward movement as you strain these fibrous gels, which I don't understand. But in the HA things, we don't see anything up and down either.
Well, if you start with a polyacrylamide gel, and you either co-polymerize with hyaluronic acid or ivermectin or double functionalize on the surface, you get the same effect.
To some extent, yeah. So we know that if we simply take a polyacrylamide gel and kind of pollute it with hyaluronic acid polymers, then the more HA we can put into that matrix, the more the matrix doesn't become much stiffer, but the cells begin to spread more. What we cannot do is get the HA density sufficiently big. If we make nothing but cross-linked HA, the density is high. The spacing between the adjacent HA change is quite small, 20 nanometers maybe. We can't quite put that much stuff in the polyacrylamide gel. But qualitatively, something like that goes on.
And last question, do you see any of the same kinds of effects with hyaluronic acid or other glycosaminoglycans?
So yeah, we do not see it with alginate. And I think years ago there was a paper from MIT or BU that had used alginate in place of polyacrylamide for chondrocyte development, and they showed really beautifully that the cells needed to get 10, 20 kilopascal alginate before they begin to spread and look like a chondrocyte. So it seems to be specific for glycosaminoglycans that cells have receptors for. Yeah, there's hey.
Just polyacrylamide with percentage the cells with J ivermectin. You see global changes on the first cells.
I think we see global effects. So one of the things we've noticed is that if we put cells on a collagen-coated gel on the bottom and an HA fibronectin gel 1 day later on the top, the cell will detach from the collagen and just move to the upper surface. So that seems to be it seems to be what's happening on one side of the cell is communicated to the adhesion receptors on the other side of the cell. Hi, Bill.
That was really great. So I think formulating this question very well. I guess what I want to know, or I'm interested in, is that in the case when you have a very stiff substrate effect, do you think that the ability of the cell to spread and organize inside the cells in the appropriate way, in that case, is of a different kind, or is that the signal now spilling over into this CD? Well, yeah. Or in other words, like, you've got a stiff substrate. Is the organization inside the skeleton perhaps CD44 dependent still?
No, I think it's not, and there. Well, we have cells from CD44 knockout animals, and they do mechanosensing fine. They have other defects. It turns out they can't polarize, but they can still spread. And they also don't completely lose this HA business either. So there are at least 6 receptors for HA, so it's not just that one alone. But yeah, I'm guessing the other thing we don't know that we're trying to look at now, even though these focal adhesions are very big, we don't know that they're stable. So they could be very large and turning over much more rapidly than the ones on glass are. So they just, in a snapshot, they look similar, but what their dynamics is, we don't know yet. But I don't think, but my guess is that you just, both of those signals, either stiffness or HA plus integrin, activate the same pathway. And once you activate that pathway, you're destined to get big actin bundles and focal adhesions. Okay, one back.
So my question is, for the HA gels that you see the cells spreading, is it just the presence of HA inducing spreading, or it comes also with, or it's coupled with optimizing tension increase? Because I would expect that when such a cell, spread a cell with so many stress fibers, if it wasn't a polyacrylamide gel 1 kilopascal, there was so much more deformation of the
Yeah. So what we know is that if we if we take one of these cells that is spread on these HA gels and we treat it with blebistatin or something, or even cytochalasin, it changes its shape very much more slowly than a cell that's spread on a stiff substrate. So I think we still probably need actomyosin activity to make any of these structures. And we know that there is actin in these actin bundles. What we don't know is how much of that actin is actually turned on. So we haven't looked at whether it's activated actomyosin or just myosin. So that's about as much as we know. We know that you cannot completely bypass pulling. You need some contractility. This isn't exactly like, this is not simply a drop wetting a surface. There's something more involved than that. But in a way, you can imagine you could get this fantastic spreading, not by putting more contraction, but just by relaxing the cell cortex and letting the adhesion energy win. So that's unfortunately that's all we know about that. But,
okay, with that note, let's finish with that part. But generally,